Regulation of gene expression in eukaryotes. Eukaryotes have high cellular diversity. The body of multicellular eukaryotes is composed of numerous types of structurally and functionally different cells. Despite this diversity in cellular structure and function, all the cells of an organism have the same genetic makeup and all of them contain the same biological information. The cellular diversity of eukaryotes therefore appears to result from differential gene expression during development and differentiation. This differential gene expression is accomplished by a complex type of gene regulation called gene modulation. Eukaryotic gene regulation is a phenomenon in which cells express only very few of their genes, nearly 15%, and different genes are expressed in different kinds of cells. This means that in eukaryotes, all the genes are not active. The active genes are different in different types of cells, and only the active genes determine the structural and functional features of the cells. So, gene regulation in eukaryotes occur at several levels such as mRNA transcription level, post-translational mRNA processing level, mRNA transport level, mRNA translation and degradation level, and post-translational protein processing and degradation level. First, transcriptional regulation. Eukaryotic cells regulate gene expression largely by regulating the rate of genetic transcription. In them, transcription usually occurs at very high rates only for rare proteins. Regulation of transcription involves different mechanisms. The commonest one among them are transcription control by promoters, enhancers, silencers, or transcription factors. Then transcription control by histones and non-histone proteins, and then transcription control by hormones and cytokines. Then transcriptional control by promoters, enhancers, silencers, and TF for transcription factor. Eukaryotic genes contain regulatory sequences that are involved in the regulation of protein transcription. They include promoters, enhancers, and silencers. Promoters, Jacob et al. In 1964, are the DNA sequences which direct RNA polymerase to its binding site to initiate genetic transcription. They interact with some DNA binding proteins called transcription factors or TF and bring about the regulation of transcription. They recognize a binding site in the promoter for the binding of RNA polymerase. Also, they recognize the short regular sequences in the promoter whose interaction regulates the initiation of transcription. Then transcription enhancers are the DNA sequences which increase the rate of transcription. So they act as the positive regulators of transcription. They are located away from the initiation site. They bind with the TF and promote transcription. Silencers are the DNA sequences which serve as negative regulators of transcription. They bind with transcription factors and repress promoter activity. This in turn lowers the rate of transcription. Silencers are specific for each cell type. They are generally located upstream of the promoter sequence. Then transcriptional control by histones and non-histone proteins. Histones and non-histone proteins associated with the chromosomal DNA of eukaryotes directly regulate genetic transcription. Probably histones repress gene DNA activity in a non-specific manner rather than selectively. They interact with the phosphate groups and stabilize the DNA double helix. As a result, strand separation becomes impossible and genetic transcription and DNA duplication gets suppressed. These histones function as non-specific repressors of genetic transcription and gene expression. Nuclear polyanions capable of complexing with histones can displace histones from DNA. This in turn promotes strand separation and thereby facilitate DNA duplication and gene transcription. So non-histone chromosomal proteins often serve as specific de-repressors to reverse this repression of transcription caused by histones. They accomplish this by binding with specific sites of DNA. This binding pulls out histones from such sites and thereby lifts the repression of transcription. Different models have been suggested to explain gene regulation by histones and non-histone proteins in eukaryotes. The important ones among them are discussed below. Frenster's model of gene-specific de-repressor RNA. Histones stabilize the DNA double helix by interacting with the negative phosphate groups of DNA. This prevents the separation of DNA strands and this prevents transcription. Thus, histones act as several repressor of protein synthesis. Gene-specific de-repressor RNA hybridizes with the non-transcribing DNA strand at a particular locus and stabilizes the loop in the open positions. This frees the other DNA and strand and permits it to transcribe messenger RNA. Then, model of specific de-repression by non-histone proteins. According to Paul and co-workers in 1971, non-histone chromosomal proteins (NHCP) can act as de-repressors to reverse or de-repress the transcriptional repression caused by histones. This de-repression is site-specific because NHCP can bind only to specific genetic loci. According to Stein and others in 1975, histones probably act as non-specific repressors of transcription. Individual non-histone chromosomal proteins recognize certain sites on DNA and bind to them. They then pull off the repressor of histones from such site, de-repressing DNA. Then, Britton-Davison gene battery hypothesis. Britton and Davidson in 1969 proposed a novel mechanism of transcription regulation in eukaryotes. According to them, hormones and other regulator molecules can control eukaryotic gene expression through the mediation of a gene battery. Some other aspects related to this will be dealt with the help of next video. Thank you.